I welcome Professor Kashyap Dubey sir. It's my utmost pleasure to actually introduce the distinguished person of this field of biotechnology to our participants, and I am really thankful to Gopalji sir for initiating this science dialogue series. Because of that, we have been in contact, or we have been at least listening to the scientists or our students or our fellow faculties, or they are able to listen, or they are able to get into uh, actually get into the point that where they can listen and interact with the such scientists or such resource persons to whom listening itself is a boon for them. So I welcome Kashyap Dubey sir. Sir, if I talk about Kashyap Dubey sir, this would be something like showing
काश्यप दुबे है
दिए को
सर.
रोशनी. हाँ हाँ, काश्यप दुबे सर, शोइंग अ दिया टू सूरज. इस तरह से अगर if I introduce him to our participant, so just going into the brief introduction for the sir. So he has completed his PhD from MDU Rohtak. Then he continued there as an assistant professor, followed by his professorship at Central University of Haryana, Mahendranagar, and then he joined to Jawaharlal Nehru University, which is number one university of our country, where he is doing a good research, a very collaborative research. He has been awarded many membership, many grants. He have been awarded, like he has been awarded membership for the National Science Academy of India in 2014. Then he have been conferred for the DST International Travel Grant for attending Oasis 2014 for the Korea, then International Travel Grant for Germany, SERB DST Young Scientist Research Grant for 2013 and 2016, and so many. So he has been awarded other accolades also, and he is a member of Biological Engineering Society, founder member since 2018. Then many others memberships are also there, for example, Association of Microbiologists of India, National Academy of Science of India, Society for the Biological Chemist, Indian Science Congress Association, Indian Society of Cell Biology 2013, and many more. If we talk about his research interest, it is basically in the field of bioprocess engineering, metabolic engineering, and environmental biotechnology, where he has published more than 150 publication in form of various research articles, in form of various book chapters and others, and he has got the citations of all these chapters and all these research articles more than 3000. So this is just a very brief introduction for his research caliber. Currently his lab is working for the development and optimization, scaling up for the bioprocesses for the production of various biopharmaceuticals, biofuels, biochemicals, and other bioproducts. So if I say that sir is an epitome for the combination of academician and the researcher, this would not be lie for us to lie. But I welcome you, Kashyap Dubey sir, and I hope that our participant will also get benefited from your vast experience and your research, and also they will get some insight from you from the top of University of India, which is Jawaharlal Nehru University. So I welcome Dubey sir for this online platform. And now, sir, I'll be handing over this virtual platform to you so that our participant can also get interaction, get insights from your research and your guidance. Thank you, sir.
Okay, so good evening, one and all. And am I audible, Gopalji?
Yes, yes, sir. Yes, sir. And
So,
I'm also grateful to you for accepting our invitation. And for participants, I would like to mention that JNU campus लोग घूमने के लिए दिल्ली में जाते हैं तो वहाँ सिक्योरिटी गार्ड होता है, लेकिन फिर भी लोग कि कोई दोस्त रह रहा है JNU में उसका नाम लेकर के, कोई जो है ना कि कोई पढ़ाई कर रहा है उसका नाम लेके, पीपल यूज टू विजिट देयर कि भाई JNU कैसा होता है. और फिर बायोटेक जाते हैं तो वहां पर में जो साइंस का उसको पता है कि बायोटेक का क्या इंपॉर्टेंस है. एंड देन डीन, हम जब जेएनयू में गए तो अब डीन से मिलने के लिए हमको लगता था कि भाई डीन से मिले कि नहीं, मिलना चाह रहे थे. सो जब हम मॉलिक्यूलर मेडिसिन में थे तो डीन से मिलना बहुत मुश्किल लगता था कि डीन से टाइम मिले कि नहीं. So I request participants to interact with him and we are fortunate that he is available, क्योंकि टॉप इंस्टीट्यूट तो जेएनयू है ही, उसमें भी बायोटेक वहाँ का सबसे जो साइंस में है, बायोटेक एंड मॉलिकुलर मेडिसिन बहुत अच्छा डिपार्टमेंट है, एंड सर इज होल्डिंग देयर administrative position also, and he is scientist, good researcher and good human being also. इनके साथ हम जो है असम में मंदिर भी गए हुए हैं, साथ में हम लोग है ना भाई की तरह, हम लोग वहां पर में वो मंदिर कामाख्या मंदिर, यस इफ आई रिमेम्बर. So he is also a very good human being. He is accessible. So I request participants, especially those who are research scholar, you can interact with him and you can take as maximum as possible from him and his knowledge. That is purpose of this science dialogue series. So I am making available to you all people, good scientists and researchers and administrator, क्योंकि जेएनयू में एडमिशन लेना एज ए स्टूडेंट बहुत डिफिकल्ट है, अब फैकल्टी बनना उससे भी डिफिकल्ट है, क्योंकि स्टूडेंट के लिए भी मुझे याद है 11 से 40 लोगों में मॉलिकुलर मेडिसिन में पीएचडी का एडमिशन हुआ, मैं अपने आप को समझ रहा था बहुत बड़ा लोग, फैकल्टी बनना फिर ये. सो विद दिस हम इसलिए बोल रहे हैं कि आप इंटरेक्ट करें, इनसे क्वेश्चन पूछें, एंड सर विल नॉट हेजिटेट टू रिप्लाई ऑल योर क्वेश्चंस, नॉट ओनली टू दिस साइंस. जेएनयू के बारे में भी अगर कुछ आपको पूछना हो तो आप लास्ट में वो इंटरेक्ट कर सकते हैं. थैंक यू सर. नाउ फ्लोर.
Okay. Yeah. So
Yes.
thank you so much, Sir Gopal, for inviting us to discuss some of the research going on in my lab for the last 10-15 years. And also I will talk about certainly about our institute. And thanks to the madam for introducing me in a very wide way. I'm not that much credential, but you know I'm still.
थोड़ा साउंड वॉइस और बढ़ सकता है क्या?
I'm अभी ठीक है क्या?
यस सर थोड़ा ठीक है.
I'm still like a student of bioprocess or biotechnology. I'm still learning many things from my student, my master's student, my PhD student, to how to do better in the bioprocess. So I will talk in my presentation from how you isolate a microbe from the soil, that means you grow in the Petri plate, then how you reach from Petri plate to the reactor.
अच्छा
So
सब
that is a kind.
पार्टिसिपेंट्स आर यू लिसनिंग हिम क्लियरली? मेरे में कंप्यूटर में दिक्कत है या सर का वॉइस कम आ रहा है? पुणे मैडम
एक्चुअली थोड़ा सा क्रैक कर रहा है, वॉइस इज क्रैकिंग.
अभी कैसा है?
एक्चुअली सर शायद आप जब सिस्टम से थोड़ा सा पीछे जाते हैं ना बैक चेयर में, तब वो क्रैक करने लगता है.
नहीं, अभी मैं बिल्कुल सिस्टम के पास ही एकदम.
हाँ
तो अभी क्लियर है सर.
ठीक है. So I will talk on the two different cases of the microbial metabolite, how you can like produce from microorganisms to Petri plate, then you go to the reactor, right? So I will talk two different cases. I will go one by one. The case one is very alarming situation. If you look at this data, I have taken from the ICMR cancer data. In the different states of our country, the number of cancer cases is very much rising. The data I am showing you a decade old. After that, many reports came. Now, in the present scenario, if you see, the data is just almost doubled and tripled. So you can think about the number of cancer cases coming into the country, and specific states like Delhi, Rajasthan, Uttar Pradesh, Bihar. If you go even in northeast area like Assam, West Bengal, Odisha, Gujarat even, Andhra Pradesh, Karnataka, like many states, they are very vulnerable towards the increasing the number of cancer cases. So what is our view? What as a biotechnologist, what we are doing, how we can help to these cancer patients, right? Another case I will discuss, that is obesity. You know, like obesity is increasing in our country. Earlier, people used to say that in rural area, people are more fit. But if you go and compare rural area versus urban area, in rural area also now people are under grip of obesity, and they are having so much lifestyle diseases like diabetes, like hypertension, and many more. So here, if you like, on what basis I am saying that they are obese? Because as per Indian Health Ministry guidelines, if the body mass index is more than 25, we will consider as a obese person. If you look at this data as comparative between men and women, you can see the men's, out of 5 men, 1 man is under extremely obese condition, and the average age is 37 years. So that means at a very young age, people are coming under the grip of this obesity issue. If you look at the data of women's, out of 7 women's, 1 woman is obese category, but at the age of 32 years. So that means if you look at the maximum risk age, that is very young age. And our country is presently going into the, like in a word, our country is the youngest, like having the most number of population, highest population in the terms of young generation. So we have to be very, very careful. So I will talk about these two issues which I have, my lab is uh, working on these two problems, two challenges. So I will start with the one that is colchicine. Okay, that is a very popular compound. Most of the biologists know, even in 10 plus 2. If you start with the alkaloid, this increasing the ploidy level, because when you use the colchicine, it changes the ploidy level into the cells. How it changed? Because when colchicine interact with the tubulin, tubulin is basically proteins. They are made up of, they produce the spindle fibers. When the cell divide, at that time spindle fiber forms. So if the tubulin polymerization is stopped, then cell will not divide. Okay, so colchicine is a chemical which is used to increase the ploidy level, right? So that is produced from the 3 different plants, and the Gloriosa superba is the plant which is in common language it is called red lily, is produced like 7.8 to 1%, and commercially that plant is exploited or explored to produce colchicine in large quantities. Here you look at the chemical structure. It has 3 ring, A, B, C. There are 5 methoxy group. If you look at carbon position 1, there is a 1 methoxy group, carbon position 2, another, 3, another, then 7, then 10. Okay, every group is having very specific activity. Okay, so I want to draw here attention. Now people start saying repurposing of drugs. How you repurpose that? Now this new word came in the COVID time, repurposing of different drugs for those you can use for COVID-19 patient. So here we are also repurposing this colchicine drug for cancer patient. Okay, so how we are going for repurposing? What are the steps? What are the challenges? I will talk one by one. Here is, you can look at the different colchicine structures. Sorry, this is the colchicine structure. In the one corner you can see, another is thiocolchicine. Here you can look at the sulfur group is attached. Sulfur group is attached. Here is the like colchicoside. Here the glycerol is attached, glycoside molecule attached. So it is called thiocolchicoside. If you look at this molecule here, the third position, the methoxy group is, methyl is removed. Here is only OH is there. But if you look at the first one, here is CH3O. Here only OH is there. So that is 3-D-methoxy colchicine. Why I am talking on this? Because when we are talking about repurposing, so first we have to target some chemical groups. Okay, on the basis of literature, we have seen that if we remove the third methoxy group of the colchicine, the compound is more potent. Potent to for what? Potent to bind with tubulin, potent to stop tubulin polymerization, as well as to reduce the toxicity. So that means if we can knock out the methoxy group at third position, the molecule will be less toxic, more potent. Okay, if that this is another molecule, another derivative of colchicine called thiocolchicine, which is used as a anti-inflammatory agent. Okay, so for anti-inflammatory drug, if we can add some this glycosidic moiety with that, then the absorption of that particular drug will be increased. Okay, so that means if the absorption power is increased, toxicity is reduced, potency is increased. On the basis of literature, we have seen that if we can repurpose both the native molecule in these two transformed or converted or the repurposed molecule, they are most effective for as a cancer drug. Now, why I am talking about this modification? Because if you look at the market size of the different naturally produced and the modified form of the drugs, look at the data here. More than, if natural products used as a drug is 14%, but 23% of the market shared by the modified form of the drug, repurposed drugs. So that means the market of the modified drugs, repurposed drug is increasing day by day. So we have to look into it, how we can convert the native molecule into the transformed one to improve the potency, improve the effectiveness. Okay, so that is the reason I'm talking on this. Now, the mechanism. What is the mechanism behind why I'm talking on colchicine? Why colchicine derivatives? Here is the mechanism of how it inhibits the tubulin. Here is the alpha beta subunits, protein subunits. There is a dimer, they form dimer, then form tetramer, then they starts growing and they are started forming microtubules. Okay, during the, which is a called spindle fiber. At the time of cell division, when the metaphase stays, then the chromosome is moving towards the different poles, then the spindle fiber starts pulling the chromosomes, right? So here is, I have compared the different cancer drugs like colchicine and the vinca alkaloids, Taxol, for to check the effectiveness. And we found that the binding strength of the colchicine derivatives with the tubulin is higher. So that is the reason we select to work on colchicine molecules. This is the whole story behind the bio process, how we reached, how we produced at the bioreactor level. This is the plant which I have shown, Gloriosa superba or Colchicum autumnale, from where you can isolate a colchicine. This is the colchicine molecule. How we could know that colchicine derivatives or the 3-demethylated one is more effective. Okay, so if a patient is taking colchicine, it is going to the liver, because liver is the organ where all the drug will go there, transform there, and whatever form is usable or effective or the active, that will go to the specific body part to cure that disease. So the colchicine is going to the this is a liver and transformed into colchicine derivative, that is the 3-demethylated colchicine. Here you can see no methoxy group is available. Okay, so what happening, we have seen that there is in liver there are so many P450 enzymes called proteins, different kind of isoforms are available. Okay, we have seen that there is a specific P450, there is a specific isoform of P450, that is C by P3A4, which is present in human liver, that is transforming colchicine derivative from colchicine to 3-demethylated colchicine. Okay, it is going to the bind with the tubulin. Okay, so that means colchicine is not that much effective. The derivatives which is going from liver to the specific site or the site of reaction, and they are more effective. Okay, so we have started working on it, like where we can found that C by P3A4 enzyme or a protein which is available. So we started screening so many microorganisms using bioinformatic approach. We have identified the protein sequence of this C by P3A4, started doing the alignment, the blast, okay, blast P. After like so many uh, alignment procedures, screening procedures, we reached at a one microorganism which is called Bacillus megaterium. Okay, here you can see the corner, that is megaterium. That is the only microbe which is having C by P3A4 present in human liver. Similar. Okay, so that means if now we can give the colchicine to in a medium and grow the Bacillus megaterium, and that enzyme is a extracellular in nature. Okay, so that enzyme will convert into colchicine into their derivative. So on that hypothesis, we started working on it. So we have isolated this microbe, developed that microbe as per our requirement, and then bio upstream process and downstream process development we started. We started growing into the sac flask. We started growing into the small lab scale 1 liter, 2 liter reactors. Okay, I will talk more on in the coming slides. And here what we are seeing that when we are downstream the or purifying the colchicine derivatives, which is transformed by C by P3A4 enzyme using Bacillus megaterium as a biocatalyst. Because Bacillus megaterium is having high content of ergosterol. So what is happening when we extract with chloroform, most of the ergosterol will extract into chloroform because chloroform is a non-polar solvent. So when we start purifying this column chromatography, the column will choke because we are purifying the using column chromatography. We are using silica as a matrix. When we start the extracted material put into the column, the ergosterol will on the top of the column, they will stop moving the column flow, they stop the flow and creating problem. So we started thinking like this is the challenge for downstream process. Can we knock out those genes which are responsible for this ergosterol production so that we can have reduce the content of ergosterol? Okay, but that was not feasible. We started working and many experiments we performed, but that was not able to like get success. Okay, so afterward we isolate the gene from the Bacillus megaterium. Okay, and then we put it into the vector and clone into the recombinant in E. coli BL21. Okay, so we clone into the E. coli, then E. coli starts growing, and E. coli now starts converting colchicine into derivatives. And when we go for downstream process, we have seen that there is a less amount of this ergosterol fought. Okay, so the problem what we are facing, that was also resolved after development of this recombinant system. So that was the whole story of the case one, which I will talk with more data in the coming slides. Here is because like in bioprocess, there is always challenge to scale up the process. So here is I have scaled up the process of the colchicine production from sac flask to 5 liter, then 70 liter. Here you can keep this data in your mind. If you look at sec flask level, when we are putting a 700 milligram of colchicine, we are getting only like 3850 milligram per liter converted one. Okay. If you look at the specific yield of the compound into the milligram per gram of dry cell weight, that is 229. If you go on productivity, milligram per liter per hour, that is 4.79. Now we scaled up. Scale up means keeping the some parameters constant. Like in 5-liter reactor level, we are having suppose 400 RPM. If we are going for 50-liter, 70-liter reactor, are we going to maintain same RPM, 400 or 500? No. Why? Because the diameter of 5-liter bioreactor is very small. If you look at the size of 70-liter, the diameter is more. If the diameter is more, the impeller is also different. Impeller size is also different. So in a 1 minute or 1 second, the movement of impeller is also different. So RPM could not be the same for 5-liter reactor to the 70-liter reactor. Why it is? Because of the diameter of the reactor as well as the size of impeller. But if you maintain these things same, then you will get more yield. Here you can see in sec flask and 5-liter yield is same almost, but at 70-liter reactor level it is increased around 500 or 400 milligram. Because in large, when you grow things in reactor, more things are controlled. You can control dissolved oxygen, you can control pH, you can control the sugar level, you can control the, if you want to add anything during the incubation time, during the reaction, you can add. So there is always a possibility to have more biomass, more yield in bioreactors as compared to the sec flask. So by this way, we increased the conversion of colchicine into the derivatives. Here I am showing you the conversion of colchicine into 3-DMC and thiocolchicine into 3-demethylated thiocolchicine. Here you can see what I just talked now. RPM for 5-liter reactor like 200 we optimized to 600. We have calculated the impeller tip velocity. What is that? Impeller tip velocity is one of the parameter for scale up. And the formula for this is pi NIDI. Okay, Ni is the number of RPM, Di is the diameter of impeller. Okay, so if you want to maintain the tip velocity constant in 5-liter and 70-liter, then you can see if here is 200 RPM and at 70-liter only 87 RPM. Here is 300 RPM, in 70-liter 130 RPM. They are very much reduced because of the diameter, because of the size of impeller, diameter of the impeller. And so this is the way of scale up. It is scale up is not like that you are growing things in 250 ml. Now you are growing in 1 liter sec flask, and people started saying that it is a scale up. It is not a scale up, right? If for a scale up, you have to maintain some of the parameters like power by number parameter, then the dissolved oxygen, KLA, volumetric oxygen transfer coefficient, impeller tip velocity. These are the some parameters which you have to maintain constant. Then you can say that it is a scale up, right? This story I have conveyed into the summary slide. We have developed, construct a recombinant E. coli for to remove the challenges at the downstream process in the form of ergosterol. So here we produce the demethylated colchicine, but when you are going for recombinant one, the conversion rate was very much reduced. Conversion rate was 55 to 60%, But if you go for the wild type one, the conversion rate was the 90%, but at the same time, the recovery, the downstream process is like recovery is 80% in case of uh, Bacillus megaterium, but in E. coli case, the recovery is more than 90%. So one side you are recovering more, one side recovery is less, one side you are getting less ergosterol, one side you are getting more ergosterol. So on that challenges we worked and we got success on it. Here is this data in tabulated form. The wild type one, the enzyme activity, what we call CYP3A4 enzyme activity. In recombinant one, you can see the enzyme activity is not that much. These are the M1, M2, M3 are the mutant which we developed after enriching the medium with the colchicine concentration like 5 grams, 7 gram, 10 gram likewise. So here you can see the biomass is less, the conversion is also less as compared to the mutant one or the Bacillus megaterium one, but certainly the overall yield is more because of more recovery rate or in recombinant one. So then we thought that because when you are using these recombinant cells, you grow the cells, you recover the product, but what happened after that? So we started thinking, can we develop a packed bed reactor system where we immobilize this recombinant E. coli into a column, okay? Then we pass through this colchicine through the column, through this packed bed reactor, and after the exit, we can get the biotransformed colchicine. So this what we have also attempted. So here you can see this is the like suppose a bead. Okay, so on bead we have immobilized recombinant E. coli. This is the scanning electron micrograph where you can see these are the E. coli cells which is immobilized. So this is basically a mathematical form. This is called uh, fixed law, where like this is the interface of the colchicine derivatives or the medium, and at the center of the point, the it always challenge because the diffusivity of the bead, if you are preparing 1%, 2%, 3% agar plate, there is always a challenge of diffusivity. There is always a change of the porosity. Okay, so here I will show you the data how we face this and how resolve it. This is the whole process what we have gone through. This is the packed bed reactor system. Here we immobilize this recombinant E. coli on the on calcium alginate wheat. Okay, so and then we pass the colchicine derivative after a holding in the column. We have maintained the flow rate, a specific flow rate. Okay, so when it comes through, then we are getting the derivative of the colchicine. Here you can look at then statistical optimization of different parameters by using a different statistical design like RSM, CCD, design of experiment, placket vermin. I will show you the detail thing. This is the whole story, how we have extracted the colchicine derivative. So here you can look at like suppose we are growing in a 5-liter bioreactor, and we are putting 35 gram of colchicine to convert into derivatives. So this is the upstream. Then the biomass, whatever coming, it goes to the treatment, effluent treatment. Then the filtered material will come as a permeate. We have to go for the solvent extraction by once or twice. It depends on the quantity available into the solvent. Then it goes to the column chromatography. After collection of the fractions of colchicine and derivative will go for crystallizer to crystallize with the help of ethyl acetate and diethyl ether. And the colchicine we have to go for recycling. So this is the whole bio process, upstream as well as downstream process for colchicine derivatives. Then what type of again solvent we can use, because all solvent are not useful, because if you want to sell your product in regulatory market, they are very specific for what we can say the residual amount of solvent available into the product. So if it is a non-polar chloroform hexane, then they will say like we are not going to buy this because of the presence of this solvent. So either you have to remove or you have to use only green solvent for extraction of these material. Here you can see the data. We have used the different or polar, methanol, ethanol, acetonitrile, then non-polar also chloroform and hexane, and we found that we are getting maximum yield in chloroform. But what to do? We are getting less yield in acetonitrile and methanol. So we started working on chloroform to extract these derivatives. At the same time, when we are putting this colchicine into the medium, cells Bacillus megaterium has to grow. So we have also seen the effect on the cell wall and plasma membrane of the Bacillus megaterium, because you know when it go into the cells, the cell wall you can see in 5 gram, in 7 gram, and 10 gram, the cell wall is also almost come finished. Plasma membrane you cannot find out. But at the same time, you can see the more inclusion bodies, the white cells, more inclusion body into cells, because you are putting cells into more stress. They are basically the carbon reserve source. When you are giving a specific selection pressure to the cells, the number of inclusion body increased. Okay, so here is also we are seeing that is increased. So that is a like in the form of spores also same. So here you can see the experiment of we immobilize the microbial cells into the calcium alginate bead. We compared this. This is the setup of the immobilized experiment we performed. This is a glass column. We put it all the these beads into the column. Okay, this is the controller of the bioreactor. So we started putting the colchicine in with the medium uh, inside this column. We maintained the rate of the pump through the controller after the maintaining the flow rate, that when the material is coming out from the column, that is converted into the colchicine derivatives. But here what is the important is, here you can reuse the cells as maximum 4 to 5 times. But if you are performing experiment in the submerged culture, in sac flask culture, where you can only use cells once. Okay, so this is the beauty of this experiment when we are started using the packed bed reactor system. But you know, like as I said in the previous slides, that that is the challenge. Like we are using chloroform as a solvent for extraction. So we started thinking that can we have some mixed solvent, non-polar and polar in different ratio to get more extraction, and say that we are using less amount of non-polar solvent. So by using statistical techniques like genetic algorithm, which is based on MATLAB software, we designed a algorithm and started these 3 solvent: chloroform, acetone, ethanol. We work out on the central composite design, then genetic algorithm further, and we found that there is a ratio of ethanol, acetone, and chloroform which are giving more extraction, more recovery. So here we can say, when we are using in the previous version 100% chloroform, but here we are using acetone and ethanol also in ratio. So we reduce the chloroform content in downstream process. So the residual content is also reduced, so that we can say that the content of this non-polar solvent reduced, so that we can easily say sell the product into the regulatory markets. At the end, like the whole story what we are talking about, that is also published in the form of review, where we put it the all therapeutic potential and mechanism action of the colchicine derivative. So that is the case 1 here ended. Okay, so in case 1, if you recall the all discussion, I started with the challenge. I started to find out the microbe. I started like using that microbe, find out the challenges, develop the recombinant, remove those challenges, started producing at sac flask, then 5 liter, then 70 liter. So that means I'm trying to show you the things from sac flask to the bioreactor. Okay. Now here is another case. I'm just taking not much time. One more case, which is obesity. Okay. So here another molecule that is called lipistatin. I will talk on it, how the lipistatin will work, because lipistatin is an anti-obesity molecule which is a competitive inhibitor of lipid, and it stops the uh, pancreatic lipase. It binds with pancreatic lipase. I will show you in the coming slide. Here you can see the data of northern India with obesity. The male and female, females are more obese than male, and the rural population you can also look at. So data is also very alarming. We have to think about the the solutions available, and we can improve the availability of these molecules to the persons, to the patients or obese persons. So here just I'm showing you in a bit that how the lipids are digested. So this is basically triglyceride, and lipase will act on it. It will form diacyl glycerol, then monocyl. So this is the mechanism of digestion lipid. So here, this is a whole story again I am showing in from one slide. So we find out that what is happening. Triglycerides is going to the converted into monoacyl, then it cross to plasma membrane, goes to the mucosal cells. It finally it is deposited into the adipose tissue. So this is the whole story in the intestinal lumen, plasma membrane, mucosal cells to deposit. So where is the key? Where is the Achilles heel? इसका कहानी आप लोगों ने सुना होगा, ग्रीक किंग का एक है ये स्टोरी, like why it is the Achilles heel was important. So we have also find out the Achilles heel of the process, that is pancreatic lipase. If we stop the digestion of the lipid here only, then nothing is going to be absorbed in plasma membrane and adipose tissue deposit. So we find out a molecule which is lipistatin, which bind with the pancreatic lipase to stop digestion of lipids. Market size, yeah, when you start working in anything, market size, you have to look into it. You can see the India is gripping into the obesity, and it is having market, very good market. Many countries, you can look at green, they are highly demanded of this obesity drug. In India, there is a several companies which are involved in R&D, not in the production, like Triveni Chemicals, Gujarat, Biocon, Roche, GSK, Alembic, IPCA. They are the main leaders, players who involved in the lipistatin production. At present, Orly State is a drug, which is a trade name of lipistatin, available into the market, which is produced by Roche and GSK. So the demand of this drug is increasing, use in the coming years. This is the molecular structure of lipistatin. This is a lipistatin molecule. This is the beta lactone ring, which is important. If something will happen with this ring, the activity of the lipistatin will goes down. When we are saying lipistatin, then lipistatin is not that much stable. So chemically, we transform lipistatin into tetrahydroxylipistatin. So you can hydrogenate it by chemical reaction into tetrahydroxylipistatin. So again, we started from the scratch. We started finding out the microbe, where it is available, how we can find out. So we have find out a microbe which is a streptomyces, which we have isolated from Haryana industrial area. We have sequenced the whole genome. Draft is available on GenBank of NCBI. We have now working on the full genome assembly. It is also just going to submit into GenBank from our lab. Just few thing important with respect to the streptomyces is the best known source for production of secondary metabolites. Very complex life cycle. That is a challenge. I'm working on it, and I will show you some data on the complex life cycle. Here is like this is the lipistatin in streptomyces, which is growing into the plate. Go for the this submerged fermentation. then separation, then you can see the lipostatin is produced. Not going into the here, not going here, you can see this is the growth of the streptomyces in the solid medium. Here you can see the mycelium. Now, this is the problem on which I am working, because this streptomyces will form mycelium, then hyphae, then pellet. Here you can see I'm showing you the three images, A, B, C. The A image is basically diffused hyphae. B image is a gram stain image. Another is C will be methylene blue stain. You can see the how the tightly woven cells, how this pellet is formed. Now the challenge is, if the product is produced at the center of the cell, center of the pellet, how it will be come out, how it can come out. What is the mass transfer limitation when substrate or the medium component will reach at the center of the pellet? Okay, so now I'm working on it to reduce, to deform this morphology, how we can beside even like change in morphology, we can put it some micro macro particles and many things and I'm working on it. So this is here, I'm just showing some microscopic images of the pellet. In the glass slide, you can see these are the pellets, beads you can see. These are basically streptomyces pellets. This is the whole cycle. It starts with hyphae, then slowly starts centralization, then mycelium, they start forming center points. So these are the 8 images. You can see how the one hyphae will start at reach at the H stage, which is a very tight form of the pellet. So it always creates a challenge for industry. So we are working on it, and this is the whole story. Then again, on the basis of genomic information, we have sequenced the whole genome. Now we have identified some of the protein network which is responsible for formation of this pellet. Now we are knocking and overexpressing or changing the expression level of these genes so that the pellet formation can be reduced. So this is the one paper what we published. Here is the story. Here you can see in better way. This is the scanning electron micrograph images. So in the A, you can see this is the tight pellet. In the B, you can see this outside of the pellet. These are the mycelial kind of things you can see. If you go in the better resolution, you can see here the small branches are producing. The small tubes you can see here. Branching, you can see the small branches are producing. So that means when the pellet starts, then after certain stage, some branching will start. So if pellet will be like even loosely woven, but after certain point of time, these branches will go inside the space available, and then tightening of the pellet starts. So that is the challenge. Okay, so from the scanning electron micrograph, I'm showing that we have then isolated the proteins of that microbe, intracellular proteins, total extracellular proteins, and we reached at a point that the proteins which are less than 20 kilodaltons are responsible, because we got some clue from another microbe, Streptomyces colicolor, which is also having similar challenge. Some researchers have identified that this DYP14A, SC by PAR-A, and FILP proteins are responsible to regulate this tip formation in the pellet. So on that clue, we started working with the Streptomyces of our microorganism. So all the protein was sequenced. We found that this is the interactome. These are the some hot spots, RPS B, DNA K. So now we are working on these points. So we have also gone for different physiological parameters, how we can change the pellet size, biomass, viscosity, oxygen, because when the tight pellet will be tight, oxygen uptake rate will also be challenged. Oxygen will not go. If oxygen will not go, how the aerobic microbes can do? It is difficult. So here I'm just showing you some data: carbon source, dextrose, sucrose, galactose, yeast. Also we tried inorganic solvent. We also tried. So this is the data what we published also. pH, we have checked the effect of pH, inoculum volume, agitation on the size of pellet. In fermentation, it is always challenge, cost of production. So we also tried to check unconventional medium component, corn flour, soya flour, sesame oil, coconut oil, and we found that in sesame oil and soya flour are getting more yield. And when we go in detail, then we have seen that sesame oil is having high content of linoleic acid, and that linoleic acid basically playing a key role in the production of lipostatic. That is the back study which we have gone. In soya flour also, we found that there are some specific amino acids which are present in soya flour, and they are responsible the formation of some proteins which are in I have shown in the interactome. These are the amino acids. So on that basis, you can see these are the unconventional source giving more yield. This is what we are working on the large scale reactors. We have gone through at 7-liter bioreactor and optimized the whole process. Here you can see at the data of the 5-liter level and the 250 ml data, you can see the yield we are getting approximately 1 gram more lipostatin as compared to the batch and sec class level data. So downstream process, as I said, that we always believe the hit and trial and the existing literature available. We have gone through the existing literature and we have modified it. Now, this is the protocol by which we are producing, extracting the lipostatin molecule from the streptomyces. These are the figures just showing you. And finally, I acknowledge two. Here I'm showing only two, but three students completed PhD on this whole two molecules, and one is in process to get PhD. And for the whole research, I got funding from SCRP, DSP, IREC. Now, friend, I thanks to the Gopalji for giving this opportunity to discuss with the particular researchers. And this is my email address you can use for any kind of communication and questions. So